hopefully you can see that right now. So I also apologize for the length of the title of my talk. I shortened it a bit, but it's still somewhat incomprehensible. But my laboratory has been interested over the years in sort of the fundamental aspects of how proteins evolve, and in particular how enzymes evolve and acquire different kinds of activity. And our model system over the last decade has really been plant systems, because they, more than any other, I think, exhibit, and we heard from Tom Mitchell-Olds yesterday, an incredible amount of chemodiversity in so-called secondary metabolism that they use to interact with and protect themselves in their environment. And my lab uses a lot of different tools. We do a lot of things. We're masters of none, but one of the tools that we really employ in the lab is the tools of structural biology to try to get a handle on simplifying sort of what we feel are the evolutionary trajectories of some of the enzyme systems that we study. I know Professor Kiesling the other night probably introduced this class of molecules known as terpenes. It's a little difficult to see in this slide, but I promise the other slides are a lot brighter. These are so-called sesquiterpenes. They're 15-carbon hydrocarbons that are made in a number of different kingdoms, but plants in particular have an incredible diversity of these carbon skeletons that they utilize, and you'll see some examples of that in a minute, for a variety of different purposes. And that's going to be the subject of my talk today. So really the question that my lab has been engaged in over the years, and this really began as a graduate student, that is, how do enzymes evolve? So in particular, we've been focusing much of our efforts on what's known as secondary metabolism, although some people in the plant world liken it to specialized metabolism. They feel it's a bit of a better name. But in particular, we're interested in how these systems emerge out of primary metabolism. So these are metabolic pathways that are highly constrained in an evolutionary setting, and then undergo a tremendous amount of expansion within a number of different organisms, and in particular plants. And what are the underlying principles that sort of drive and guide that kind of evolutionary change. So this is an example of the pathways that I'm going to be talking about today, and I believe Jay introduced this the other night. On the upper left here is the so-called mevalonate pathway that you and I have and that we share with the cytoplasm of plant systems. And this is a primary metabolic pathway. It's essential. And ultimately what it produces through 5 carbon building blocks is this very critical molecule shown here known as farnesyl diphosphate. It serves as at the pathway or the intersection of a primary metabolic pathway to make a number of essential compounds in all species, as well as in plants, fungi, and in bacteria, but mainly in plants. This molecule also can serve as a substrate for downstream specialized metabolic pathways to make molecules like this. And the molecule artemisinin is derived from a molecule of this pathway through so-called secondary metabolism. I've also illustrated on this slide a comparison about this issue of emergence out of primary metabolism. This is an essential enzyme in all organisms, farnesyl diphosphate synthase. There are variations of it that build different kinds of carbon building blocks. And it has an all helical fold and an active site center shown here, and it uses metal ions to actually link 5 carbon building blocks together that are the downstream products of the mevalonate pathway, and also the so-called MEP pathway that's found in certain bacteria as well as in the plant plastid. But the interesting thing is, a few years ago, our lab solved the structure of the first plant sesquiterpene synthase, and it actually had a conserved fold with this primary metabolic enzyme FPP synthase, and that's what's illustrated here in gold. It also had an active site that consisted of 19 amino acid residues that line the active site surface. And Jay's group has done some really lovely work in directed evolution in narrowing the substrate or the product specificity of one member of this family. But in general, plants have a large number of genes that encode these enzymes. They clearly have an ancestral link with primary metabolism, but instead of making FPP, in this particular case, they use it as their substrate. They ionize off the diphosphate linkage, and then they can direct that resultant high-energy intermediate down a number of different pathways to create these kind of hydrocarbon molecules. And so we've been probing, in a phylogenetic sense, what are the key elements within this fold that give rise to the diversity of products that we see in these metabolic pathways. So just very quickly, I just want to go very quickly through specialized or secondary metabolism to give you an introduction to it. You've probably heard something about this from Jay's talk. These are pathways that are not necessarily essential, but they really do provide a great measure of fitness in plants. And Tom Mitchell-Olds talked about this with glucosinolates in Arabidopsis the other day, or Arabidopsis cousins. So these are compounds that really do have an essential role when a plant is out in nature in its natural environment. It's a language system. So the molecules, including the ones I'm going to talk about today, are utilized for attraction, repulsion. Sometimes they're toxic, sometimes they're not. In some cases, they're used for screening a number of abiotic factors in the environment. And so these are pathways that get switched on when a plant in a native ecosystem undergoes both either biotic or abiotic stress responses. Very often, if you look at these classes of molecules, at least the products of them are often unique within a set of related species. And so there may be some contribution in some sense to speciation. We study these pathways from a fundamental point of view because it's like evolution on steroids. And I'll show you examples of that in a few slides. So that in relatively short evolutionary timescales, you have substantial functional shifts in these systems. So from a linear biology point of view, annotating these various pathways is quite difficult because even with a few changes, and I'll show you even cases of SNPs that result in non-synonymous mutations in these proteins, you can have substantial functional shifts. Our lab is also interested in this issue of mutational drift protein evolution, and we find that studying secondary metabolism is allowing us to address a question that is widespread in the community, but I don't think is generally applicable, and that is that the majority of missense or non-synonymous mutations in proteins are deleterious to the stability of the proteins. That's clearly the case in systems that we've studied in primary metabolism, but in secondary metabolism, even though there's a common ancestry of these systems, these proteins seem to be much more tolerable to missense mutations and therefore more evolvable. And then finally, these pathways have a translational component. They really are the reservoir and inspiration for the majority of clinically prescribed drugs. You heard from Jay the other night about artemisinin. It belongs to this family of compounds. So not only in the pharmaceutical community, but these molecules can also be of importance in the bioenergy field, and there are a number of people that are exploiting these kind of molecules from that standpoint. So I'm going to focus the rest of my talk on this class of proteins known as terpene synthase, or some people call them cyclases, because the majority of the compounds that are made by these proteins in plants are of a cyclic nature. And really the idea is, in a very simple-minded way, is to use the tools of structural biology to link this area of space, primary sequence information, with this chemical output that we see on the right. And that is, can we at some point get to a point where we can actually predict with a high degree of specificity this primary sequence information, how it links to this great diversity of small molecules that are made. Now, these are small. They're about 204 Daltons in size, but they're highly constrained molecules with a number of stereocenters, very complex cyclic structures, and that cyclic structure of these products is imparted in some way by the diversity of sequence that we see in the terpene synthase enzymes. And I'll show you how we can exploit that information. So the model system that I'm going to speak about today comes actually out of the Nicotiana. We're going to talk about a plant that has a somewhat infamous connotation. This is Nicotiana tabacum. And I just wanted to illustrate here that this is what has brought my group into the sequencing world, and that is that this is a very interesting genus, and the genera that surround it are also quite interesting. They belong to the family of plants known as Solanaceae, quite important plants from a standpoint of human biology and agriculture. Even within Nicotiana, there are greater than 70 species, and something that I find quite intriguing from a geological standpoint is that the emergence of the Nicotiana genus and its distribution now is quite interesting. It seems to coincide with the movement of the continents. So in fact, the largest number of species and where this particular genus was supposed to have emerged is in South America, but the second largest is actually in Australia. And it's thought that at one point when Australia and South America were linked through Antarctica, the so-called Antarctic effect, that there was distribution of the species. And now we have a number of Nicotiana species that have been isolated in Australia for quite some time, and so that it provides a very interesting genus to probe the evolution of particularly chemodiversity, which has a role locally in local ecosystems. Okay, so here's our metabolic model. It's probably one of the simplest metabolic pathways that one could think of. In the case of the tobacco plant, as well as a number of Solanaceae, including things like tomato and potato and eggplant, etc., when a fungal spore or pathogen lands on the plant, they mount a very complex defense response. And one of the things that occurs late in that process is the upregulation of two genes, and in the case of tobacco, a gene that encodes an enzyme that is a terpene synthase known as tobacco epiaerostolachin synthase, and then a downstream P450 that has a bit more substrate promiscuity associated with it. The key step is this TOS protein that now takes this so-called primary metabolite, FPP, shuttles part of the carbon out of primary metabolic pathways, ionizes this pyrophosphate, and then in some way directs the cyclization of that resultant chemical intermediate into this bicyclic product known as 5-epiaerostolachin that then is hydroxylated sequentially by this P450 in a stereospecific manner to make capsidiol. And capsidiol is a so-called phytoalexin. It's not cytotoxic against these pathogenic fungus, it's cytostatic. So what it does is it essentially keeps the fungal spores from spreading over the plant and limits damage to the plant system. And so this is the metabolic pathway that we've been studying. These are also enzymes that are quite amenable to functional characterization. So we have a medium throughput system where we use a combination of gas chromatography and mass spectrometry to actually analyze these systems both in vivo as well as in vitro. And this is an example of an in vitro reaction of that tobacco enzyme, epiaerostolachin synthase. And from this you can see that there is one major peak, and this is our epiaerostolachin peak. And the mass spectrometer is quite useful because depending upon the chemical structure of the product, they'll undergo very specific fragmentation patterns in that spectrum. And from the work of a number of laboratories around the world and wonderful fragmentation libraries, this actually can fingerprint, at least to a rough approximation, what the identity of that small molecule product is. We can't actually determine the stereochemical features of the molecule, but we can often place it within a particular class. And so with a relatively facile experiment, we can actually fingerprint the products that are being made, and in addition, with the appropriate use of standards, we can quantitate these systems and we can derive from them, for instance, steady-state kinetic constants, and understand not only what the product diversity and substrate specificity promiscuity of the system is, but also quantitatively understand the catalytic efficiency by which they operate. So if you look at this spectrum, though, you can see a number of minor peaks along this spectrum. And this is indicative of the whole family of terpene synthases. And in Jay's case, he exploited one member of this family that was particularly adept at making a whole host of minor products, some of which equaled in intensity the major product in the spectrum. In our particular case, about 80% of the time at ambient conditions, the major product is epiaerostolachin. But if you look closely at this minor spectrum, it turns out that they also are sesquiterpene hydrocarbons. And in fact, their presence depends on the presence of an active enzyme. And I'm not going to go into the chemistry, but the nature and the chemical identity of many of these products elucidated through scale-up reactions and analytical chemistry can be rationalized based on what we think the chemical mechanism of this particular enzyme is. But in the rest of the talk, what I want to do is focus your attention on these four compounds and tell you two stories. One that's a little bit of an older story and one that has just appeared in print that has to do with this issue of substrate promiscuity, that is the ability of these systems to actually access more than one substrate, and this issue of product diversity, that is the ability of these enzymes often to produce many products, and how they link together through what I call mechanistic elasticity. That is the ability of a single enzyme in a single active site to actually move carbon or move intermediates down different mechanistic pathways. So for the first part, I just want to focus your attention now on this compound and this compound. Here's our major compound, epiaerostolachin. If you look at this compound, epiaeromorpholine, which constitutes about 10% of the spectrum, you'll notice that it has the same carbon skeleton and stereochemistry of the major product, but the position of this double bond is actually different. It's in a different regio, so it's a stereo isomer. It's in a different position. And in fact, the position of that double bond is shared in many ways, and I'll make more light of this in a minute, with this product, primnospheridine, shown here. So if you align these two ring systems, you appear to have in this epiaromophylline molecule a so-called chemical hybrid. It has characteristics of this major product and characteristics of this minor product, and I'll tell you why I think that's important in the next few slides. So the first is that there turns out to be a phylogenetically related plant, the Solanaceae. It belongs to the Hyoscyamus genus, or the so-called henbanes. And this is Egyptian henbane, Hyoscyamus muticus. It actually has, it actually has an enzyme in its system known as henbane primnospheridine synthase. It shares 80% sequence identity both at the gene and protein levels with the enzyme from tobacco. It utilizes the same substrate, farnesyl diphosphate, but instead of making epiarostolicine as its major product, it now makes that primnospheridine product that was a minor product in the spectrum that I showed of the tobacco enzyme. And given this, we felt that this was a nice model system to try to dissect what are within the 20% sequence variation between those two, what is it that actually is helping to specify the particular mechanistic pathway that these two different enzymes diverge at. Okay, so here's the Hyoscyamus genus, or clade. It's actually very closely related to Nicotiana. So somehow in that sequence variation between the two is encoded the ability to control the kind of carbon skeleton that you produce at the end. So one of the questions that you may ask is, are these things so different? And the answer to that is no, because they actually probably transit through a common chemical intermediate. Promise to limit the chemistry that I talk about today. But where they differ is in the final two steps of the mechanistic pathway. So there are a number of steps that they share in common to get to this common high-energy chemical intermediate. And where they differ is that the Hyoscyamus enzyme actually allows for a methylene group migration instead of a methyl group migration. And then the reaction is quenched through the loss of an acidic proton in the case of Hyoscyamus at position 6, and in the case of the tobacco enzyme at position 8, and that's where they diverge. And that epiaromophylline product that is a minor product in the tobacco spectrum that I said is a so-called chemical hybrid of the two, it actually shares the carbon skeleton migration with the tobacco enzyme, but the deprotonation machinery of the henbane enzyme. So it diverges at those final two steps. So we ask a very, very simple question. Using this structure, what is it about the sequence variation in and around the active site? So out of the 20% sequence variation, it's about 100 amino acid differences between the two proteins. We needed a simpler system to probe this in an evolutionary sense. And if you look in and around the active site, there are actually 9 differences between the henbane and the tobacco enzyme, 2 of which at this position 516 and 402 that lie on the active site surface, and 7 of which lie in the surrounding tiers. So at first thought, we thought we had the key switch, which were these active site changes at these 2 positions. And this is likened to the work that Jay has done on 19 amino acids that we defined earlier on in our crystal structures to actually encompass all the regions within the active site that would touch substrate or intermediates in the reaction trajectory. However, if we make those 2 mutations in the active site, we do very nicely and very cleanly convert the enzyme, but not into a primnospheridine synthase. We've now made an epiaromophylline synthase, and that's what's shown here in this purple color. So the double mutant in the active site positions was not sufficient to switch the specificity or the product diversity to that of the henbane enzyme, but now we've made an enzyme that for all intent and purposes is making this so-called chemical hybrid. So with those 2 active site positions, we took the 2 final steps in the reaction and we've now separated them. So we retained the carbon skeleton migration chemistry of the tobacco enzyme, but we have the deprotonation machinery that is shared with the henbane enzyme. So an interesting result. Now if we go beyond the active site and we make the 7 other additional changes, so these are residues that are not, at least from the static crystal structure, directly in contact with the substrate or intermediates during the reaction pathway. So they form like layers of an onion, a surrounding sphere of residues. And now we're able to cleanly convert the enzyme into a primnospheridine synthase. So this is the tobacco enzyme with those 9 mutations. And if we characterize the kinetic constants of that enzyme, it's actually identical to that of the wild-type enzyme. So not only do we convert the product diversity profile of the enzyme, but retain the catalytic efficiency of that process. And we can also go in the other direction. So if you take the henbane enzyme and you make either 8 or 9 mutations, we're able to take the henbane enzyme and convert it into an epi-aristoleucine synthase, again, without paying any penalty in terms of catalytic efficiency. So it's very clear that not only is the active site important, but layers that surround the active site. And we have new data taken from NMR data and limited proteolysis that really dynamics are playing a very critical role in this family of proteins in directing the kind of chemistry or the kind of products that are made in these particular settings. But then we wanted to ask another question, and that is, what are all the possible permutations, assuming this simple model that these 9 mutations are what's necessary and sufficient for this functional shift? What are all the possible mutational pathways that really separate those two enzymes? And if we could then recapitulate that in an ancestral sequence reconstruction kind of way, do transitions between these two extant sequences involve enzymes that are much more promiscuous or diverse in their product profiles? And ultimately, we wanted this is not directed evolution, we want to base our model on phylogenetic information. And this is the work of a former postdoc of my lab, Paul O'Malley, who now has his own group at John Innes, who's really took on this task and has done a spectacular job. So if we look within 15 angstroms of the active site, we have those 9 positions that I just mentioned. And what we really wanted to do is look at all the mutational pathways that might connect from some common ancestor, and that's a factorial, 9 factorial. So that means there are about 360,000 odd potential pathways of mutant combinations that would traverse from a common ancestor. Very difficult problem, but we could actually look at nodes on the pathway, so that's a simpler number, 2 to the 9. So if we looked at all possible combinations of mutants between these two enzymes, that's 512, and that's what Paul's actually done. And so I'm going to show you the results of that analysis where we've produced all these mutants and isolated them and carefully characterized them. And then as a teaser, we've just now finished the corresponding library in the background of the henbane enzyme. We don't have a lot of functional data on that yet, but we have now both libraries in both directions constructed. So here's the results of the tobacco enzyme library, and this is a space-filling tetrahedron that I'm showing here. And I'm just color coding things based on the percentage of a particular product made. So if a mutant actually lies within this quadrant, it's an epi-aromophylline synthase. This quadrant, it's wild type. Here it's promiscuous. It doesn't make anything at greater than 50%. And here it's a primnospiridiene synthase. And as we expected, the majority of things are promiscuous. About half of the library is promiscuous. 90% of the library is active. But we do have a number of these other activities that are shown in the library. And I want to just sort of, this is a busy slide, but show you some examples of this. So this is, I've turned it sideways just so it's a little bit easier to see. But this is a collection of GC traces where we've now color-coded them like in the previous slide. So blue is wild-type activity. This pink color is epi-aromophylline activity. Light blue is promiscuous activity, and red is primnospiridiene synthase activity. And together with Gerard Manning, who's in the audience, we were able to use a very simple mathematical method to encode the information within this slide, and then traditional neighbor joining algorithms to build what we call a chemical clade that will cluster mutants based on how similar they are in terms of their product spectrum. And so while we have a lot of promiscuous mutants within this library, what I want to highlight, though, is that they have very distinct features. So the ratio of one product to the other is shared within this clade, but not shared with this clade. That becomes important because it's known in some plant systems where it's not a single compound that actually has the bioactivity. It's actually a ratio of one compound to the other. So encoded within this library is the ability to make very specific mixtures of compounds that may be useful for various bioactivity profile characteristics. So about 90% of those let me go back. One more thing. So while most things go through this promiscuous intermediate, we do have some things that exhibit what we call saltatory changes. So if you look at this mutant, this is an M7. Okay, so we've made 7 mutations in this mutant, but it's retained wild-type activity. There's a SNP that's non-synonymous, and we now flip the enzyme into a primnospiridiene synthase. So here's a case which is somewhat reminiscent of punctuated equilibrium or a saltatory change, where a single nucleotide polymorphism, albeit in the case of this somewhat artificial reconstruction of these ancestral sequences, is able to skip and bypass all these promiscuous intermediates and quickly flip its product spectrum into that of the other compound. So as I said, 90% of the mutants are purified at high levels. This was unexpected to us. We felt that we would get, as we expected, most missense mutations would have a deleterious effect on the protein. In this particular case, that's not true. Most mutations were approximately additive in the effects on product output. On average, it took about 2 mutations to go through this promiscuous intermediate, but we also, in about 10% of the cases, have these so-called saltatory changes where you can have very rapid shifts in the product spectrum of a particular enzyme. Okay. So with the final 3 minutes, I just want to touch on one final story, and that relates to this particular compound here, known as prezazyme, and this has to do with substrate promiscuity. Ignore the chemistry here. I just want to focus your attention on this starting material, FPP. It has 3 double bonds in it, and the stereochemistry of the double bonds is quite important. So a natural farnesyl diphosphate that we all make is all trans, and so we have a trans and a trans double bond. There are a class of terpene synthases, one of which that Jay studies, a morphodiene synthase involved in artemisinin biosynthesis, that actually, once they ionize this particular carbocation, can allow for a isomerization around this double bond into a cis pathway to produce a large amount of cisoid-derived products. However, when we looked at the minor product spectrum of the tobacco enzyme, we found a compound that was derived from the cisoid pathway, and we felt that was a little unusual. And so together with Bob Coates' lab at the University of Illinois Urbana-Champaign, we synthetically made a substrate that had the cis configuration. And lo and behold, when you feed this so-called non-natural substrate to the enzyme, you get the product spectrum shown here in red. And that is, it no longer produces the normal product, but now we produce this tricyclic prezazyme product. And in fact, the chemical features of that, the enzyme features of that, it's actually a little bit better substrate than the natural substrate in producing this particular product, which is a bit unusual and something that's selective of some kind of enzymatic trait. So then we used crystallography to kind of define what is it about the active site that would allow this enzyme to actually go down these 2 seemingly very disparate pathways. I should also say that this is not a property shared with other terpene synthases, so if you give this non-natural substrate to other terpene synthases, including our henbane enzyme, they don't touch it. The tobacco enzyme seems to have this unique ability to utilize it as an alternative substrate. So with crystallography, we were able to show that at least the static X-ray crystal structure, the active site volume, is conducive to go down one of either of those 2 pathways. We could use computational chemistry to model the energetics of these intermediates. Again, they fit within the active site confines, again explaining why the enzyme can transit depending upon the starting material down 2 separate pathways. And here are the final products, again consistent with the active site structure elucidated by X-ray crystallography. So we're kind of left with this question, so what? Is this biologically relevant? So just 2 things to mention in passing. One is that FPP synthases, it turns out that our so-called non-natural cis-trans FPP substrate isn't so non-natural. If you analyze almost all FPP synthases, it turns out that 5 to 15% of the time, they actually make the cis product in this particular case. So there is the distinct possibility that there is a pool of cis FPP made in a whole host of organisms that could be used in a manner like we see in this particular case with this tobacco enzyme. The only thing I could say finally is that we have to go back to the plant to try to understand what's actually going on. I don't have a slide here because we've just done the experiment, but we've collected volatiles from the leaves of tobacco. And in fact, what we find is that cis-derived product, prezazyme, forms a major component of the volatile emission from the leaves of tobacco. So it does appear that in the plant itself, these products are actually being made and emitted from the leaves. We don't know what role that they may be playing, but in many cases, the leaf volatiles, particularly of the sesquiterpene hydrocarbon class, are used to mediate a variety of different insect-plant interactions, whether it's to repel herbivorous insects or to even attract predatory insects that will feed on the herbivorous insects. And so that's something that we have yet to look at. So with that, I will stop. I've already mentioned a number of people that have contributed to this over the years throughout my talk, and I want to acknowledge HHMI for actually supporting the work, and thank you all for staying around.
I had a quick question. You did very elegant work showing the mutations that you put in these proteins, and the two species had 80% identity or something. Yeah. Can you trace back in a real phylogenetic sense to when events occurred, and were they clustered, or was it just a stepwise progression?
So that's a great question. So this is what has brought our labs into the sequencing arena. There is no information of other Nicotiana species in terms of this particular family of proteins. And so we're now growing in our laboratory over 50 different species of Nicotiana, and we're doing deep transcriptome sequencing on them to actually, so that's the next question, is to really ask, so what did nature actually do? Great question.
What's known about the structural organization of the enzyme complexes or the storage products?
Oh, so the whole issue of subcellular localization. It's a fantastic question. So first on subcellular localization, it's something we're now beginning to look at. If you noticed in that crystal structure, there's this blue domain that I didn't talk about. That is a plant-specific domain. So the family members that are found in fungi and bacteria only have that catalytic orange domain. They lack that other domain. That plant-specific domain shares structural homology with endoglucanases, ironically, but it doesn't have the catalytic residues of the endoglucanases. So we think it's probably involved in some form of subcellular targeting. We think that's also important because it's known, when I got back from a meeting in Israel in November, and there are two pools of FPP in the cytoplasm. There's a pool that goes into primary metabolism, and there's a pool that seems to be sequestered and used only for secondary metabolism. So I think understanding where this enzyme is and at the appropriate time will give us a lot of information about what's going on. Detlef.
Joe, how do your results transitioning from one activity to the other compare to what Huddle has found, converting one enzyme to another and the evolutionary paths that can be taken? Do you come to similar conclusions there?
I think the one thing that we observed, that a number of people observe, is this whole issue of transiting through a promiscuous kind of intermediate. So we see things like that. I think the thing that we're not observing, though, is the sheer number of mutants that retain wild-type levels of activity and stability. Now, I should qualify the stability issue because we are interested in protein evolution. And so what we're using now is a qPCR machine from Roche, actually, not for qPCR, but actually to look at the thermostability of these proteins. What we know so far in the extant species, that the thermal stability and the activity profile of the protein is linked with the native environment in which that plant is found. So the henbane enzyme, which is found in the Middle East and in hotter climates, much more stable enzyme. And now we're characterizing the mutant libraries, and the stability profiles of the mutants are varying depending upon the extent to which we mutagenize it. But I think what is a bit surprising is the number of successes out of the library, which is about 90%. So that is different than what others have.